But in the meantime, hi everyone, I'm McDonagh. I'm Translational Informatics Director at Open Targets. Brilliant, and we've got the slides up. So what is Open Targets? So we are a partnership between our academic institutes, the EBI and the Wellcome Sanger Institute, and currently 5 industry partners. So that's GSK, Sanofi, Pfizer, Genentech, Merck, MSD, and together we're a partnership to help transform drug discovery through the systematic identification and prioritization of targets. And our approach is really to help build this evidence between targets and diseases or phenotypes using data that we generate through our research program, as well as that we integrate and analyze from publicly available data. And why are we interested in functional genomics? So we're really keen to understand more around disease mechanisms, so going from variant to gene to function to phenotype, and context-specific function. So really understanding the function of targets in cell types, across different tissues, in disease versus healthy. And that helps build this evidence base between what's going to be the best target to modulate, and we can start to develop therapeutic hypotheses from that. So functional genomics can give us ideas around understanding the directionality and the effect of modulating a target, for us to understand whether that would be effective in treating the disease and also safe and not cause too many side effects. And to give you a quick two examples of across this axis, of where we generate new data is the IBD-verse projects that we fund, and another project that we funded that helps integrate publicly available data is the perturbation catalogue. So I don't have time today to run through the IBD-verse project, but really excitingly, early June, a Nature paper came out from that project and a Nature Genetics paper last week. And we think this is the largest that we're aware of single cell collection from patients. And these papers both help reveal cellular context of disease mechanisms in IBD, and have also identified potential novel disease targets. So go and check out those papers. And then the perturbation catalogue we funded, it's an EBI available resource that's available through that URL. And this helps capture perturbation data across both variant, gene and expression level, across different modalities, and they've currently got over 1,000 data sets now in there for you to utilise. So go check it out, but also go check out Poster 34 today for more information around EBI resources. But I wanted to focus today on how our core team help bring together data that we generate through our projects, our research projects, and integrate publicly available data, and do this with genetics and functional genomics data at scale. So why is genetics evidence important for drug discovery? There's a huge attrition rate, potentially lots of new medicines, but only ultimately one gets approved because lots fail in clinical trials due to safety, and safety as well as efficacy, lack of efficacy. And Matt Nelson's group recently published that drug mechanisms with genetic support have 2.6 greater probability of success. And interestingly, that actually we're far from reaching peak genetic insights to help discover new targets for more effective drugs. As well as that, actually, side effects are twice as likely when the target gene carries genetic evidence for a similar trait. So for us, we really wanted to know for a given disease, what are all the genes that are associated with that disease, so they could be potential efficacious targets, and can we use genetic evidence for that? But also, having an aspect of the universe for a given gene, the universe of the diseases and traits that that is associated with, so that we can start to understand whether it might be safe to modulate that target, or would you start to cause other phenotypes. So GWAS gives us an opportunity, a huge wealth of genetic evidence data. However, it's really hard to go from a genetic signal from GWAS to the likely causal gene underlying that association with that trait or disease. Often these signals are from variants that are non-coding, and often the lead variant might actually not be the causal variant due to linkage disequilibrium. And how do we do this? Across scale, across all diseases and traits and GWAS data. So our solution has been to create open targets gentropy. So this is a Python package for post-GWAS analysis that's automated and modular, really to help address this issue of ever increasing bigger data sets and more complex statistical analysis that we want to be able to do to go from those GWAS signals to likely causal genes. So this is just a schematic of the gentropy pipeline, high level. And I'll walk you through this. So we work very closely with the GWAS catalogue at the EBI to bring in GWAS from their resource, as well as FinGen, UK Biobank GWAS. We undertake fine mapping to generate GWAS credible sets. Then we work closely as well with the EQTL catalogue, another EBI resource, who provide us with molecular QTL credible sets. So that's eQTLs, single cell eQTLs, pQTLs, and splice QTLs. We undertake co-localization analysis across those data sets. So we do GWAS to GWAS co-localization and GWAS to molecular QTL co-localization. And this all feeds in to this feature matrix for a machine learning pipeline that we called the locus to gene scoring pipeline. So the ultimate goal here is to help provide a predictive score for the most likely causal genes underlying each of those GWAS associations for each trait and disease. And this is very much done at scale. So just to give you an idea of the numbers here, we bring in over 100,000 GWAS studies, 2 million molecular QTL credible sets. We have now over 800,000 GWAS credible sets from that, ultimately resulting in more than 500,000 locus to gene score predictions, so target disease or target trait associations. And from this at scale analysis, we've been able to create a map of all the human gene disease trait associations that we see from this GWAS data. So across more than just over 9,000 diseases and traits from GWAS studies, we see that there are 8,285 genes that are associated with disease, and around 15,000 genes associated with complex traits. And this pleiotropy is pervasive across the genome. So pleiotropy is when a particular gene is associated with more than one disease or trait. So we've shown that 64% of disease genes are pleiotropic, so they're associated with more than one disease. And of those disease genes, 57% of them are also linked to more than one therapeutic area. We've also shown that we as a community are still discovering new associations from GWAS. So here's the year on the x-axis here, up to 2024. And you can see for gene pleiotropy score, so the number of unique diseases per gene across all GWAS is continuing to grow. We're discovering more diseases for the same genes. and the variant pleiotropy score as well. So the number of unique diseases per variant is also growing. And we're discovering, more variants per gene for disease associations over time. And we also reiterate that genetic diversity is really important for this novel discovery. So here again, on the x-axis, we have year over time. And this plot shows disease-associated genes, which are increasing over time as we make new discoveries. And then this is unique gene disease pairs that are being discovered over time from GWAS data. So this is the year of the study of the GWAS. And we see that actually around 50% of those unique gene disease associations that we're discovering are from studies where there's more than 10% non-European ancestry participants in there. So it's really important. We're finding more new discoveries thanks to this increase in non-European ancestry studies. But ultimately, we've got this nice map of all the associations across the genome for disease and traits, but how can this actually help us prioritise targets systematically? And that's ultimately what we want to do. So for drugs that have gene disease pair association, that would be developed for gene disease pairs that have protein altering variation support, we found would be much more likely to be approved and successful. However, genes that do have the protein altering variation support from GWAS are also much more likely to be more pleiotropic and have high pleiotropy and be associated with side effects. So these genes were enriched in cancer driver genes, they're enriched in adverse reaction toxicity screens, and things like mouse knockout lethal genes. So they're also more likely to be unsafe to target by a drug. So we realised that actually what you want is gene disease pairs that have genetic evidence for protein altering variants, but also an intermediate pleiotropy score. So not too high because that's unsafe, not too low because you've not got enough association with disease. So these, this is the framework that we came up with for, identifying which gene disease pairs might be the most likely to succeed clinically. And these have an odds ratio of 10. And using this criteria, we find that 50 out of around the 242 drug target indication pairs that are approved, 52 of those have this criteria already, but we were able to find 2,700 pairs that are potential targets that have this criteria. They have medium level pleiotropy plus protein altering variant support. And we think that these could be optimal for drug targets. So you can read more, I haven't covered everything, but we have a preprint out around this work. But ultimately, that all then feeds into the Open Targets platform, which is our open source resource for prioritising target disease associations and really helping build therapeutic hypotheses. So here you can build a comprehensive picture of your variant of interest. So we bring in functional context for around 7 million rare and common variants. And this includes associated phenotypes from gentropy fed through and clinical disease annotations, as well as pharmacogenetic annotations. Then you can also see whether your variant of interest, what are the different variant effect predictions for that, nomad annotation, and we've also brought in ENCODE ETG, which helps provide annotation on whether that may be a regulatory enhancer variant, as well as being able to see your variant in the context of the protein structure and play around with that, see which domains that's in, and whether it's in likely pathogenic region hotspot. So that's variants, but what about actually prioritizing the best targets for a disease? So here's an example of a disease page. So for Alzheimer's, we have on the left-hand side the list of genes that have been associated with this disease, coming from evidence from different data types. And the darker the blue, the more weighting of evidence for that association between that gene and disease. So here we see the evidence that's coming from our Gentropy pipeline from GWAS, but also alongside that, gene burden and rare disease clinical genetics evidence, such as from PanelApp, somatic mutations, evidence from whether there's known drugs for this target disease association or clinical trial, perturbation and pathway analysis, literature, expression, and animal model evidence. And this is fully dynamic, so you can actually upload your own gene list if you have a screen and you want to understand the evidence for those genes. It will automatically populate that for you. You can define the weighting of those evidences and choose which data types you want to include, and that will reprioritize your target lists. You can filter by biological pathway, for example, or go term, share this with colleagues, and then immediately dig, by clicking on the circle, dig into the underlying evidence and the original sources for that. And also look at whether there are molecular interactors for your targets of interest that may have more evidence for association with that disease. But we go beyond this, so I'm just looking at the time, we go to help you make decisions like, has this target been drugged before? So this is our framework traffic light system, where red indicates unfavourable attributes of a target, and green is green for go, maybe more favourable attributes. So you can also ask, would a small molecule chemical probe be available to move forward with validation studies? Would a small molecule antibody PROTAC, sorry, I skipped that slide, be more suitable modality for that target? And how safe might it be to actually modulate that target? And we've just updated this yesterday in our June release, so you'll be able to see tissue specificity, distribution update, but also cell specificity and distribution. This is all open source, so you can query this by the web interface, but also download everything or query via a variety of ways. And we're just to move on to how we're collaborating with the UK Functional Genomics Initiative. So for example, for the protein post-translational modification cluster led by Matthew Child, this involves the PropVar resource at MLEBI, led by James Stevenson up there, where they will be feeding in the missense variant functional annotation as part of the cluster work. We actually link to PropVar, to and from PropVar, so we hope to integrate that variant annotation into the Open Targets platform, and then that's available with all the other variant annotation that we provide. We're also collaborating with Kenny Bailey's group, who is part of the molecular mechanisms cluster. So they're generating single cell molecular QTLs from patients, and we hope to feed this into the Gentropy pipeline and feed back to the group, fine mapping, all the co-localization and those target to disease associations that we find from that. And then once they're happy, once that's published, we will push that out to the public within the Open Targets platform. And then the Data Coordinator Centre here have been working closely with EMBL EBI groups to understand where there are already data standards, workflows, pipelines that could be utilised by the clusters, and what repositories would be ideal for depositing that data, making it available for the wider community. So go and see poster 34 today to find out more. So overall, just to summarise, we're having to create this ecosystem that helps us get insights from functional genomics to inform on how we develop safer and more effective drugs. So both by generating new data that's more context specific, bringing in and integrating publicly available data and providing this across platforms, repositories and pipelines. And a big thank you to our team at Open Targets, all our partners and collaborators, and obviously the participants that make that data available so that we can glean these insights. And thank you very much for listening. Thank you. Happy to take questions.